Hello students, welcome back to the next part of antigen antibody reactions. In the last lecture, we have studied some conventional reactions of antigen and antibody. I want to remind you that we have finished the precipitation, agglutination and complement fixation reactions which are known to be under conventional reactions of antigen and antibody reactions. And now we are going to start with some newer techniques, that is radioimmunoassay, ELISA, that is enzyme linked immunosorbent assay, and immunofluorescence. These are called as newer techniques, but which are also based on antigen antibody reactions. Now, what do you mean by immunoassay, or what is immunoassay basically? And why do you need these newer techniques? In agglutination and precipitation reactions, antigen antibody complexes are large enough to be seen by the naked eyes. Whereas if these reactions, that is antigen and antibody, these are reacting, but if not visible, then the agglutination and precipitation techniques will not be useful to us. Then in such a cases, what are the techniques that we will be using? Now, what are the reasons when we will not be able to see the reactions between antigen and antibody? The reason might be the antigen and antibody are present in very low concentration, that they are reacting but not forming a very large complexes, that they are visible enough to form agglutination and precipitation. And that is the reason we are not able to see those complexes. Now in such a cases, you won't be able to see the visible changes in vitro, that is in the microplate or in the test tube. That is, you won't be able to see agglutination and precipitation reaction, and you won't be able to quantify antigen or antibody. For example, it means when you have a very low concentration of antigen or antibody, the agglutination, precipitation or complement fixation techniques won't be able to help you. In such a cases, you need some advanced technique to quantify antigen or antibody when they are at a lower concentration. So, to determine such a low concentration of antigen or antibody, the techniques used are immunoassay. There are basically commonly used two types of immunoassays: radioimmunoassay and enzyme-linked immunosorbent assay, that is ELISA. These both the techniques are extremely sensitive. For example, the radioimmunoassay can detect the antigen concentration as low as 0.001 microgram per ml. And ELISA can even detect the concentration in picogram per ml. Meaning to tell you that both of these techniques are extremely sensitive and can detect the concentration of antigen or an antibody at a very low concentration. So now let us start looking at, in this lecture, we are going to more concentrate on radioimmunoassay. Now both these technique, that is radioimmunoassay or ELISA, they are nothing but labeled or tagged immunoassay. Now what do you mean by labeled or tagged immunoassay? Basically, Why we call them as an immunoassay? You have learned in analysis, assay means finding out the concentration, that is finding out the quantity. So immunoassay is a biochemical test that measures, just a minute, that measures the concentration of macromolecule or small molecules like hormones, drugs, or proteins via antigen antibody reactions. So assay means measuring the concentration. Why you call them as immuno? Because it measures the concentration of either hormones, drug, or proteins, but via antigen antibody reaction as an immunoassay. Then both RIA and ELISA, that is radioimmunoassay and enzyme-linked immunosorbent assay, both are called as labeled or tagged immunoassay. Why you call them as labeled or tagged immunoassay? Now let us see that. This is an, say suppose, well, a micro plate well, and this is an antigen, and this is an antibody. So antibody is specific to antigen, and this antibody will bind to the antigen. And this specificity we are using to find out the concentration of molecule through antigen antibody reaction. But problem is, as we are saying, the antigen is binding to the antibody, but the reaction is not visible to the naked eye. And that is the reason, to make this reaction visible, we need to put some label. We need to tag something. Now this tagging can be done either to the antibody, or we can even tag, or we can even label the antigen. So tag or a label is attached to antibody or antigen so that previously unseen reaction due to low concentration can now be seen because of the presence of tab or label. So you can see now there was no tab or label, so this reaction was unseen. But this time, this antigen and this is an antibody. Now you can put a tag or a label to antigen or antibody. Here is an example. The tag or a label is added to the antibody. When this antibody gets attached itself to the antigen, now because of the presence of label or a tag, you can find out the concentration of this complex, or either an antigen or an antibody. This tag can be a radioisotope or an enzyme. If this tag is a radioisotope, you call this method as radioimmunoassay. If this tag is enzyme, you call this method as ELISA, enzyme linked immunosorbent assay. I hope everybody is very clear with what are labeled or tagged immunoassay, and that is why RIA and ELISA both are labeled immunoassay. So in this lecture, we are going to see details of radioimmunoassay where the tag is radioisotope. So basically, radioimmunoassay is where label uses radioisotope and radiation emitted is measured by gamma counter. An example of radioisotope is I-121. Whereas ELISA Label used is enzyme. Then substrate is added to indicate binding of antibody to antigen. So you can see the label used here is enzyme. And to check whether the antigen is bound to the antibody or not, you add the substrate, which is substrate for this enzyme. And this substrate then convert this substrate is converted into the product, and because of the presence of a product, you understand the antigen and antibody is bound. This enzyme converts substrates to form a color product. Example is horseradish peroxidase. These are the enzymes ka example. And you can also have alkaline phosphatase as a enzyme. Substrate for horseradish peroxidase is OPD, that is O-phenylene diamine dihydrochloride. And for alkaline phosphatase, substrate can be p-nitrophenyl phosphate. Anyways, we are going to see ELISA again in detail in another lecture. Right now, let us concentrate on radioimmunoassay. So basically, radioimmunoassay, the labeled use is radioisotope.